So we have talked about the DNA replication, and now let us see how does this very long DNA get packed into the nucleus of a cell, which is so microscopic. How long is the length of the DNA? Let's analyze this. The length of the DNA double helix in a typical mammalian cell is approximately 2.2 meters. Can you imagine? आपका एक सेल और आपके एक सेल के अंदर न्यूक्लियस ऐसे 100 ट्रिलियन सेल्स होते हैं आपके बॉडी के अंदर एंड सेल के अंदर न्यूक्लियस में से डीएनए निकालेंगे इट हैज अ लेंथ ऑफ 2.2 meters. That is the length of the DNA. If you remove all the DNAs and 100 ट्रिलियन cells के DNAs निकाल के if you put them in a straight line, it covers a distance from the earth to the moon. That is the length of DNA found within one human body. Can you imagine what a microscopic structure it must be, so thin that we have Earth to Moon ka distance packed in the form of small ultra thin DNAs found in the nucleus, each DNA having a length of 2.2 meters. How did we come to this length? How did we come to this length? Take the distance between two consecutive base pairs as 0.34 nanometers. If you remember, we had just talked about it. What is the length of the DNA? Sir, ये हमने last lecture किया था. This is the pitch of the DNA. And pitch of the DNA was 34 angstrom units. It had in it 10 base pairs. 10 base pairs. So distance between two base pairs was 3.4 angstrom units. 3.4 angstrom units is same as 0.34 nanometers. 3.4 angstrom units is same as 0.34 nanometers. So the length between two consecutive base pairs is 0.34 nanometer, which is same as 0.34 into 10 raised to minus 9 meters. Now, how much DNA is present, or how many base pairs are present in one haploid DNA? एक haploid DNA, we remember last lecture we had done 10 raised to 3.3 billion base pair, 3.3 into 10 raised to 9. So this is haploid. So diploid करेंगे, it is another 3.3 into 10 raised to 9. So 3.3 into 10 raised to 9 maternal DNA and 3.3 into 10 raised to 9 paternal DNA. So दो DNA के threads है हमारे cell के अंदर. So that is 3.3 into 10 raised to 9 maternal, 3.3 into 10 raised to 9 paternal. That is a total of 6.6 into 10 raised to 9 base pairs. So how many base pairs we have? 6.6 into 10 raised to 9 base pairs. So we have 6.6 into 10 raised to 9 base pairs, and एक base pair का distance है 0.34 nanometers, which is 0.34 into 10 raised to minus 9 base pair. 10 raised to minus 9, 10 raised to 9 get cancelled. 6.6 into 0.34 turns out to be 2.2 meters, and 2.2 meters का DNA is found in the nucleus, which is 1 micron. 10 raised to minus 6 मीटर का न्यूक्लियस होता है, एंड एक माइक्रोन का न्यूक्लियस होता है, उसमें 2.2 meters का डीएनए अकोमोडेट करते हैं. How can this long DNA be accommodated in such a small nucleus? For which the DNA is highly condensed and coiled and super coiled to fit into the nucleus. डीएनए को कंडेंस करके कॉइल और सुपर कॉइल करके न्यूक्लियस में डाला जाता है. हाउ इज दिस पैकिंग डन? In eukaryotes, the packaging or organization of DNA is rather complex. डीएनए को पैक करने के लिए histone proteins are required for packaging of the DNA. Histone proteins. Histones are proteins that are rich in basic amino acid, lysine, and arginine, which carry positive charge in their side chain. तो समझो एक हिस्टोन प्रोटीन है. अ प्रोटीन इज अ चेन ऑफ अमाइनो एसिड. अ प्रोटीन इज अ चेन ऑफ अमाइनो एसिड. तो हिस्टोन प्रोटीन is a chain of many amino acid: methionine, arginine, serine, lysine, arginine, lysine, serine, histidine, glutamic acid, arginine, lysine, arginine. तो ज्यादा करके कौन सा वाला अमाइनो एसिड रिपीट होते जा रहा है? आर्जिनिन और लाइसिन, आर्जिनिन और लाइसिन, आर्जिनिन और लाइसिन, आर्जिनिन और लाइसिन. तो ये ज्यादा करके लाइसिन और आर्जिनिन अमाइनो एसिड है, which are basic amino acids, and they are positively charged. And DNA by itself is negatively charged. So, negatively charged DNA is having an electrochemical attraction, an electrostatic force of attraction around the positively charged histone proteins, and DNA gets wound around histone proteins. 8 molecules of histones, 2H2A, 2H2B, 2H3, and 2H4 get organized to form something called as a histone octamer or a core. तो एक हिस्टोन कोर के अंदर दो H2A मॉलिक्यूल्स है, उसके पीछे दो H2B मॉलिक्यूल्स है, उसके नीचे 2H3 मॉलिक्यूल्स है, उसके पीछे 4H4 मॉलिक्यूल्स है. समथिंग लाइक अ क्यूब. 2H2A, 2H2B form a square. 2H3, 2H4 form a square. Two squares are superimposed over each other. It forms a cube. 2H2As, 2H2Bs, 2H3s, and 2H4 का एक क्यूब होता है, and that is called as a histone octamer core. Histone octamer core over which is present the DNA. और डीएनए का 1/3 टर्न्स होता है. डीएनए का 1/3 टर्न्स होता है. तो समथिंग लाइक आपके पास एक क्यूब है of octamer of histones. You take 2 cotton threads and turn it. तो आपने cotton threads को ऐसे turn कर दिया. So 2 cotton threads look like one, and you pick up one histone core and take one turn, and take 3/4 turn, leave a little space, take one more histone core, take one turn, take 3/4 turn, leave a little space, take one more core, one turn, 3/4 turn, and leave a little space. So, sir, 1/3 जो cotton thread है is DNA. Double stranded DNA का 1/3 turn is there, little space, 1/3 turn is there, little space, और वो 1/3 turn डीएनए निकल जाएगा ऑक्टोमर से. तो उसके ऊपर हम लोग समझो एक च्युइंगम चिपका देते हैं, तो कॉटन थ्रेड चिपक जाएगा. दैट च्युइंगम इज अ H1 molecule. Chewing gum which sticks the 1/3 turn is an H1 molecule. So we have octamer of 8, 1/3 turn, H1 molecule. A little space, octamer of 8, 134 turn chewing gum, that is H1 molecule. Octomer of 8, 134 turn of DNA and H1 molecule. And this is the way it is present in the form of a bead string. Bead string. This is bead. This is string. This is bead. This is string. This is bead. This is string, called as a classical bead on a string appearance. Bead on a string appearance. And sir, we have octomer of 8, उसके ऊपर 134 turn of DNA is called as core DNA. Core DNA has 146 base pairs, and adjacent nucleosomes are having small segments of DNA which are called linker DNA, which are 54 base pairs. तो सर ये दो H2A है, दो H2B है, दो H3 है, दो H4 है, उसके ऊपर 134 टर्न्स ऑफ DNA है, is called as one nucleosome. इसे एक न्यूक्लियोसोम कहते हैं. It has got adenine, thymine, thymine, adenine, adenine, thymine, guanine, cytosine, cytosine, guanine, guanine, cytosine, cytosine, guanine, guanine, cytosine, cytosine, guanine, guanine, cytosine, adenine, thymine. ऐसे base pairs हैं. This is A pair with T, T pair with A, G pair with C, C pair with G, A pair with T, T pair with A. ऐसे टोटल कितने बेस पेयर्स है? You have a total of 154 base pairs. When 154 base pairs, I'm sorry, 146 base pairs. 146 base pairs are present in 134 turns. In 134 turns, you have a total of 146 base pairs. तो ये 134 टर्न में कितने बेस पेयर्स है? A total of 146 base pairs, and that is called as the core DNA. इसे core DNA कहते हैं. And one bead on string with H1, one bead on string with H1, one bead on string with 134 turn of DNA with H1, one bead on string with 134 turn of DNA. How many base pairs are present on the 134 turn? 146 base pairs. यहां कितने हैं? 146 base pairs. और उनके बीच में होता है DNA, which is called as linker DNA. What is that DNA called as? Linker DNA. And linker DNA has 54 base pairs. Linker DNA has 54 base pairs. So one bead and string has a total of 146 plus 54 base pair is a total of 200 base pairs. 200 base pairs. So एक बीड के ऊपर कितने हैं? 146 base pairs, which is core DNA, and string, which is called linker DNA, has in it 54 base pairs. So a total of 200 base pairs are present. तो एक core DNA में 146 base pairs and linker DNA has 54 base pair. So the nucleosome has a total of 200 base pair. So if we look at the thread again, this is bead string, bead string, bead string, bead string, bead string, bead string, bead string. How many turns of DNA here? 134 turn, 134 turn, 134 turn. How many base pairs in 134 turn? 146 base pairs. And what is that called as? The core DNA. और बीच में string is 54 base pairs. String is 54 base pair. and that is called linker DNA. So totally how many base pairs? 146 plus 54 is a total of 200 base pairs. So how much is this? 200 base pair, 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 200 base pair. And that is the way 200 base pairs are found in one nucleosome. ऐसे कितने बेस पेयर्स 200 बेस पेयर्स you have to find out how many beads are there. How do you calculate how many beads are there? This is some homework for you. If you remember, you have 3.3 into 10 raised to 9 base pairs divided by 200 base pairs. That will give you the answer of how many beads are there in one single chromatin thread. तो एक सिंगल क्रोमैटिन थ्रेड में कितने बीड्स है करो डिवाइड 3.3 into 10 raised to 9 divide by 200. That means if two zeros are gone, this will become 10 raised to 7, and 3.3 divided by 2 will become around 1 point. Do the maths. How much will that be? 1.85. 1.85 plus 1.85 is 5 and 5 10, and 8 and 8 16, and 1 17, and 3.7. So that becomes a little higher. Just do the maths. You will get around 1 point. How much would that be? 3.3. So 1.5, 1.65. 1.65 plus 1.65 is 5 and 5 10, and 6 and 6 12, and 13, and 3.3. So around 1.65 into 10 raised to 7 beads are there. 1.65 into 10 raised to 7 beads are there. That is 3.3 into 10 raised to 9 base pairs divided by 200 is 10 raised to 7, and 3.3 divided by 2 is 1.65 into 10 raised to 7 beads are there in one DNA. In one DNA. ऐसे दो DNAs है, maternal and paternal. So it is 1.65 into 10 raised to 7 beads per DNA are present, or per bead on string appearance are present in the entire packing. And third, these nucleosomes are repeating units of thread stain colored bodies called as chromatin, which is called as the beads on a string appearance. The string or the DNA is 2 nanometer or 20 angstroms. The DNA by itself is 2 nanometer or 20 angstroms. And 20 angstrom. That we had done. DNA का diameter कितना होता है? 20 angstrom. The beads on a string appearance called chromatin fiber is condensed into nucleosome fiber, which is 10 nanometers or 100 angstroms. तो ये आपका chromatin fiber कितना होता है? 100 angstrom. This is one chromatin fiber. ये DNA कितना होता है? 20 angstroms. DNA कितना होता है? 20 angstroms. And one bead कितना होता है? 100 angstroms. 100 angstroms or 10 nanometers. This is one bead, and this is string, and then another bead. This is 100 angstroms, which is called chromatin fiber. And ये है 100 angstroms, 100 angstroms, 100 angstroms, 100 angstrom, which is bead on a string. What will happen during packing? This we have just done it in cell division. Chromatin fiber gets condensed into nucleosome fiber, which is same as chromatin fiber, 100 angstrom. ये याद है हमने किया था bead string, bead string, bead string, bead string, bead string, bead string, bead string gets packed into bead, 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 string, 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 which is called nucleosome fiber. And that will be 10 nanometer or 100 angstroms, which will then get packed into a telephone wire, which is called as a solenoid. So which will get packed into a telephone wire-like appearance, which is called as a solenoid. And that is solenoid fiber is 30 nanometer or 300 angstroms. So how much was bead, 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 string, 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 nucleosome fiber? Nucleosome fiber was 100 angstroms or 10 nanometers. And how much is bead, 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 telephone wire like condensed structure solenoid fiber is 300 angstroms or 30 nanometer. It will get packed further and further and further to form chromatids, which form a chromosome made up of multiple beads. And this is a bead, 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 heavily beaded structure. And these are called as chromatids. And these chromatids are about, if you check that carefully, 700 nanometer. 700 nanometer is one chromatid. So one chromatid is 700 nanometer. This is 700 nanometer. This is 700 nanometer. So how much was bead string, bead string, bead string, bead string? 10 nanometer. How much was bead, 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 string, 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 string? 10 nanometer. So chromatin fiber was 10 nanometer. Nucleosome fiber was 10 nanometer. How much was bead, 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 like a telephone wire solenoid fiber? It was 30 nanometer. And how much is bead, 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 bead? That is chromosome or chromatin fiber, 700 nanometer. So 10 nanometers has become 30 nanometers, has become 700 nanometers. That is the way packaging takes place. And packaging of chromatin at a higher level requires additional set of proteins which are called as non-histone chromosomal protein. At an additional level, more packaging takes place. And more packaging takes place, and you have ये आपका क्रोमोसोम मेड अप ऑफ क्रोमैटिन, ज्यादा बीच में है सेंट्रोमियर, साइड में है काइनेटोकोर्स, and centromere and kinetochores on the side are made up of proteins which are called as non-histone chromosomal proteins. So those proteins are called non-histone chromosomal proteins. In a typical nucleus, some region of the chromatin is loosely packed and stays light and is called as euchromatin, which is transcriptionally active chromatin. तो ये है आपका एक्चुअल क्रोमैटिन फाइबर. तो क्रोमैटिन फाइबर ऐसा दिखता है. You have bead string, 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 और ये है bead string, bead string, bead string, bead string, bead string, bead string, bead string. Similarly, यहाँ पर है bead string, bead string, bead string, bead string. तो some of the bead string is tightly packed and some of it is loosely packed. तो जहां बीड स्ट्रिंग बीड स्ट्रिंग बीड स्ट्रिंग बीड स्ट्रिंग लूजली पैक्ड होता है एंड इट स्टेन्स लाइट इज कॉल्ड एज euchromatin. And this chromatin undergoes transcription and translation. In other words, it forms protein and it forms protein and is tightly packed. It is forming protein and is loosely packed. And you have heterochromatin, which was darkly packed, is called as heterochromatin. It is densely packed and stains dark and is transcriptionally inactive. तो ये transcriptionally inactive होता है, which is heterochromatin, which is densely packed. So lightly packed is euchromatin, which is transcriptionally active. Darkly packed is heterochromatin, which is transcriptionally inactive. This was the packaging of the DNA.